designing the therapy, and then prevention. So how to define? Antibiotic should be used, which route of administration? Most of us, we go for intrauterine, but I have some difference of opinion into that. First 30 days of postpartum, please do not go for intrauterine. It is not going to help you, but it is going to harm the animal. Whatever the thing that you are administering. But it is better to avoid, because having 4 or 5 animals getting conceived in this process and harming another 50 animals is not going to help. So first 30 days or at least first 21 days postpartum, intrauterine should be avoided. I have my personal experience with buffaloes as well as cattle. So that is the best way. Go for systemic routes. So why some animals do not respond? This is also a $1 million question, and the differential response. I will come to that. So therapy, I am not going to recommend that these are all the therapy. This is the thing, if you use 100%, you are going to get. But mostly based on the literature, what is available, this seems to be good. So there are 5 pillars. This applies to uterine infection as a general or suspected subclinical uterine infection affected repeat breeding animals as well. First prong is eliminate bacterial infection. Systemic is used in case of postpartum immediate, and uterine infusion maybe after 30 days. Improve uterine contraction, this is a must. Ecbolics are necessary, and supportive care. Recurrence prevention is very, very important, because whatever the treatment that we are giving in this particular cycle, make the animal to come into short cycle, so that herself is equipped with the immune system, so that she herself clear all the things and ready for insemination. So recurrence prevention is very important, and then immunity enhancement, it is a supplementary for other therapies. So based on all the collected literatures, these are 3 conditions. If it is endometriosis is there, it's a general, I'm not talking only repeat breeding. Endometriosis, cefepime benzoate, that is told to be, it is quite good, and levofloxacin, metronidazole, ornidazole, all other things. But the success rate is around 80 to 90% with this. And one more thing is, give a TGF2 alpha later parts, maybe after 7 days or 8 days. Make her to come into heat by her own. Make her to come into short cycle. So that is the best way of treating the subclinical uterine infection suspected repeat breeding animals. And then there are some alternate therapies. These alternate therapies have been tried extensively by several groups. Several groups says this is good, but some groups says it's not that much. So whatever the thing that is collected, I have given LPS, it is well proved. Lipopolysaccharide administration of this much is going to be good. Ludhiana means Gaduvasu has done commendable work on this, and they have recommended this one, but unfortunately it is not being used in many places. Then oyster glycogen, cholesterol, the cholesterol whey, normal cholesterol, animal cholesterol, and then take the whey only, and then intrauterine. This activates in the C3b complementary way, and then helps the animal to eliminate serum. These are all the things that is something different, uterine immunity modulation. But these reduction of bacterial population, they are gaining a lot of momentum now. And some of things are really promising. Essential oil, and then dextrose. Dextrose, 50% reduction in the uterine infection. Just administration of 200 ml. Dextrose says it reduces the bacterial growth and increases the uterine tone. And then it also nurtures the endometrial cells. So it helps in the 3 directions. So, but a lot of studies are not conducted, but whatever the studies they have been conducted, just administration of dextrose in the subclinical animals, and they get good clearance. And then similarly, in case of ozone, 50 ml of ozonized intrauterine distilled sterile solution, this has been found to be very good. And now researchers at SAUs, many SAUs, they have taken up this research. So it's also showing a promising way, these 2 dextrose and then ozone. And herbal formulation, there is a big list, and whatever the thing I could get with a convincing results in 3 or 4 studies I have given here. Neem oil, 25 ml intrauterine for 3 days. Mostly IVRI and other areas they have done this. And then Tinospora, this is I think Uduchi. So 50 ml intrauterine for 3 days, garlic extract. They all claim that it is going to be beneficial for eliminating the uterine infection. And this is the last question, why some animals cannot clear? Here comes very important, that is called negative energy balance. I told you, you see this animal and this animal, and if we give GnRH here and here, do you expect the same response? Cannot. If somebody expects the same response, then they have to understand that negative energy balance is very critical. So this animal is under NEBAL. Negative energy balance, it is the one thing which makes the animal non-responsive to the antibiotic and highly responsive or susceptible to the uterine infection. So negative energy balance, I will be explaining how to prevent that in later. So negative energy balance, one have to take care of it. And then this is very recently emerging. in case of animals, but it has already been reported for over 2 decades in case of human being. One is bacterial biofilm formation. We are giving antibiotic, but the animal is not at all responsive. And if you see that in vitro, there are some inhibitions, but in vivo it is not. Why it is? So in that case, this bacterial biofilm formation is one important thing that prevents the entry of the antibiotic into this. So what happens here? This is planktonic form. Planktonic form means individual bacteria. Because all the antibiotic, most of the antimicrobial treatments available are generally developed and evaluated against microorganisms of planktonic, means free living. But here it is not free living. That's how the antibody is not able to act. So what happens here is, in the first, this bacteria moves and colonizes in some area. When the environment is quite good, these extra polymeric substances, these substances are released, and then this biofilm getting matured. And once fully matured, there are a lot of bacteria into that. And after that, the microcolonies are released so that they go to the other area and start infection, spreading infection. And what it does, why it is important here? Because of this one. The antibodies, these extra polymeric substances, they prevent the antibodies from entering into the colony, so that they will not get into these and they will not be able to act on the bacteria. And these are all some of the persistent cells. So they carry this antibiotic resistance. They are the very important cells which are responsible for spreading this antimicrobial resistance. So this biofilm is potentially dangerous, one in terms of antimicrobial resistance, two in terms of non-responding the antibiotic therapy. So what we can do? So this is one of the study conducted by Dr. Liba. She is from Kerala. So what they could find, in generally it is said that N-acetylcysteine, hydrogen peroxide, tris plus disodium EDTA. So these three are very good. They can chelate and they can remove this biofilm. They can eradicate this biofilm and they can help in attenuating the bacterial growth. So in that case, what is found was 90 to 100% clinical recovery in those animals which are administered with NAC or in combination NAC with antibiotic. So this is one thing, to me, it's promising, that can be taken in the field conditions. Like tris plus disodium, this we have used when I was there in NDRI in 1997 to 1999. We also tried with this tris disodium EDTA along with the antibiotic, and that gave us good results.